So how do we group or classify organisms today? Well, we have several different ways, and for several different reasons. We have a common name and a scientific name. And these are important based on where we are, what we're doing. Common names we use in everyday life, and they're regionally important. Scientific names are more specific, and they're globally important. They're in Latin, and you always write the genus and the species. You make sure to capitalize the genus and lowercase the species. Underline if you're handwriting. If you're typing, you can underline or italicize. But you have to set it apart. How do you find the name of organisms? Well, you use a dichotomous key. And here we have a dichotomous key that shows us how to find the common name of an organism. So we have an organism with feathers, and we want to find the name. So, does our organism have feathers? Let's say it does. So, yes. Does our organism swim? Let's say it does. Yes. So we follow it along. Follow it along until we find out that our organism is a duck. Dichotomous keys are also available to give us scientific names of organisms. Now we're going to look at some history of classifications. You all have been doing, you've looked at the domains. There were 3 domains. History, though, Aristotle classified organisms in 2 ways, as plants and as animals, simply because that's what he could see. And then that was about 2,000 years ago. And that's what you would about expect, because that's what he could see. Then during about the Greeks, Roman times, they started to group the animals and the plants into smaller groups. And that's where we found genre. And the singular for genre is genus. And then about the Middle Ages, we started to see even more grouping within the genres, and we found species. So that leads us up to our current taxonomy, or class system of taxonomy. That began with Linnaeus, who used a polynomial system. And polynomial is similar to what we're familiar with today, where we have many names for organisms. If you were to not just give the scientific name, but if you were to give the whole name, you would start at the kingdom and go down kingdom, phylum, class, order. I mean, you would give kingdom, the phylum, the class, the order, the family, the genus, and the species. So for the polar bear, you would have Animalia, Chordata, Mammalia, Carnivora, Ursidae, Ursus, Arctos. And so Linnaeus found that very, very cumbersome. So he developed a shorthand, which was a binomial, which of course is 2, which leads us to our current system of scientific names, of scientific naming. Which is what we just talked about, the genus and species. Again, we have to underline or italicize, capitalize genus. But let's look at the kingdoms a little bit more in depth. Excuse me. So you were talking about the domains, and the 3 domains were archaea, bacteria, and eukarya. Just a little review. So now let's look at our kingdoms a little more in detail. So we have cell type, and we have prokaryotes and eukaryotes. Nuclear envelope is absent and present. Transcription occurs in the same compartment for prokaryotes, or for the archaeobacteria and the bacteria. For eukaryotes, it occurs in different compartments. The cell wall in archaeobacteria, it's present. It's also present in bacteria. For Protista, it's present in some forms. It's present in fungi in the form of chitin. It's present in plantae in the form of cellulose, and not present in Animalia. For mode of nutrition, we have berries for archaeobacteria and bacteria. And we have berries here with Protista. It's heterotrophic for fungi, autotrophic for plantae, in heterotrophic or in a male. And then the last category that we have there is multicellular. For archaeobacteria, that category is absent because they're all unicellular. It would be absent for most of the Protista, but not all. It's present in fungi because fungi are multicellular, and present in all forms of animals and plants.